Hello everyone, welcome back to my channel. This is Saroj Parajuli and today I will be talking about RNA-I, or commonly known as RNA interference. We can also call it gene silencing or suppression of gene. In the last three videos, we talked about the structure and composition of RNA and different types of RNA 1 and different types of RNA part 2. And if you want to watch these videos on RNA composition and also the types of RNA, I will put the link in the description section and you can watch these videos from the link provided. And in this video today we are going to talk about the RNA interference. Before the discovery of genome editing tools like TALEN and zinc finger nuclease and also the CRISPR-Cas9 genome editing tool, RNA-I was very popular. However, after the discovery of those three genome editing tools, zinc finger nuclease, TALEN and CRISPR-Cas9 genome editing, RNA-I got less popular. However, RNA-I is still very important if we want to work on silencing of a gene. So before RNA-I, there was a tool called zinc finger nuclease, and this is also a genome editing tool. And another tool is TALEN, TALEN, and another tool is the very popular tool CRISPR-Cas9. So we are not going to talk about these three genome editing tool in this lecture. However, we will talk in the future lecture in detail. So we will have each section for zinc finger nuclease, TALEN and CRISPR-Cas9 genome editing tool. Today in this lecture, we will only focus on the RNA-I gene silencing or RNA-I suppression. So if we look at this figure, in the nucleus there is the production of RNA molecule. So sometimes double-stranded RNA molecule can form, and this is a double-stranded RNA molecule, and this is a hairpin loop. So this is a double-stranded RNA molecule. We'll mainly talk about four different steps how the RNA silencing occurs. In the first step, there is a formation of double-stranded RNA molecule, either with the hairpin or without the hairpin. In the second step, there is an enzyme called dicer. It cleaves those RNA molecule into small pieces. And in the third step, the protein called argonaut carries the single-stranded RNA molecule along with other proteins to form a structure called RNA-induced silencing complex, which is commonly called as RISC. And in the fourth step, in the presence of ATP molecule, those RISC complexes are activated. And after activation, this complex is attached with the RNA molecule and it cleaves those RNA into small pieces. And this is how the silencing occurs. So this is just brief introduction. So now let's talk about those four steps in detail. Before starting these four different steps, I'll talk about some background. So RNA is transcribed in the nucleus, inside the nucleus, and the DNA is being transcribed into transcript, or we can call it RNA or pre-RNA. So this is done by RNA polymerase II enzyme. So this is a DNA molecule, and DNA molecule is being transcribed into RNA molecule. And what happens, this RNA molecule has different structure. So there are exon, intron, exon and intron. So what happens, RNA splicing and the maturation of the RNA occurs, and all the introns are removed and the only exons are ligated together. These transcripts move in the cytoplasm. So this is a transcript, and what happens, now the protein synthesis occurs. So in RNAi, what happens if there is a double-stranded RNA, then this is going to bind to this transcript, and this transcript will be cleaved and silencing occurs because this transcript cannot make the protein and it is degraded and it makes the silencing. There are so many transcripts and some of them are being degraded. This is called RNA interference or RNA silencing or RNA suppression. The first step in RNA interference is the formation of double-stranded RNA. So there are two different types of RNA that can do silencing. One is siRNA and other one is mRNA. So we will talk about the detail in these two RNA and what are the difference and what are the similarities between these two RNAs in next video. However, we'll just talk about the common RNAi suppression method in this video. Now, if there is a formation of double-stranded RNA either by siRNA or by mRNA, then this is a start of the RNAi. So RNAi occurs only after the formation of double-stranded RNA, and these double-stranded RNA molecules are identified by an enzyme called dicer, and dicer cleaves those RNA molecule into small pieces. So usually it cleaves those RNA molecule into 21 to 23 base pair. So this is the primer for RNAi suppression, and this is step 2. Now let's talk about the step 3. In step 3, after dicer cuts those double-stranded RNA into smaller size, 21 to 23 base pair in size, then the argonaut protein, it identifies the double-stranded smaller fragment of those double-stranded RNA molecule, which is 21 to 23 base pair in size, and this argonaut protein, it will identify it and it will take those double-stranded RNA molecule, and one of the strand which we call as guide strand, so we can call this one as a guide strand. So the guide strand will be associated with the argonaut protein. However, the other strand, and sometimes we can call it as passenger strand, and the other strand gets degraded, and this guide strand along with argonaut protein and some other proteins forms a complex, and this complex is called RISC, and this is commonly called as RNA-induced silencing complex. So this is the main complex that is responsible for RNA silencing, and this guide strand will guide the So what happens now? This guide strand is responsible for guiding all those RISC complex to the particular site. So it will guide this complex to the complementary DNA sequence or complementary RNA sequence, and the silencing occurs. So this is the step number 3 or third step. Now let's talk about the step number 4. In the fourth step, the activation of the RISC occurs by the presence of ATP molecule. So the ATP molecule will activate the RISC complex, and it will be able to cleave those RNA molecule. So the guide strand will identify the complementary base in the messenger RNA, and it will pair with the messenger RNA. And after pairing with the messenger RNA, what it will do? Now it is going to cleave this mRNA fragment into smaller size, and hence protein synthesis doesn't occur and RNA silencing or RNA interference happens. These are 4 different steps of RNAi for gene silencing or the suppression of RNA. And now let's talk about how activated RISC can silence a gene. So it can silence a gene in 4 different ways. The first one is the cleavage of messenger RNA or mRNA. So what happens in this step, the endonuclease of the RISC complex will chop the mRNA into smaller pieces, and it cannot make the protein, and the silencing occurs. And the second step, its second way. So in the second way, it will block the translation. The RISC complex will attach to the mRNA molecule, and instead of chopping the molecule into smaller pieces, it can block the translation of mRNA to amino acid and stop the formation of protein. And third way how it silences a gene is binding to the DNA molecule in the nucleus. So the argonaut protein will move into the nucleus, and instead of binding to the RNA molecule, it binds to the DNA molecule, and it will do the silence. So how it does silencing? So it will attach to the DNA in the nucleus, and it will recruit some other chromatin remodeling complex around the promoter region. So this is a gene region, and this is a promoter region. So the RISC will attach to this gene region because it has complementary sequence, and this complex will recruit some chromatin remodeling complex in the promoter region, and this can help in suppression of a gene. Instead of RNA, it works on the DNA by itself. And the fourth way is the RNA synthesis to generate the double-stranded molecule. So what happens here? So this is a RNA molecule, and it attaches somewhere here, right here. The RISC complex will attach here, and instead of chopping the RNA molecule, it does the transcription. So it does the transcription by RNA polymerase enzyme, and it makes the double-stranded molecule. Now this double-stranded molecule can make a precursor for RNA silencing, and the cycle repeats again from the beginning. Today we talked about RNAi, gene silencing or suppression of mRNA, and we also talked about the different ways how a RISC complex can silence a gene or RNA. So in next video we'll be talking about the RNAi applications, practical applications of RNA. So till then, thank you so much for watching this video, and if you like my content and want to learn more about molecular biology, genetics, genomics, genome engineering, and more on bioinformatics, please like my content, share my content, and subscribe to my channel. Thank you so much for watching this video, and I'll see you in next video.